Thanks, Jeff, for the introduction. Alrighty, so let's get into this then. And before I get started too, I just do want to really acknowledge Michigan Wheat Program for checkoff support. Without that, we really wouldn't be able to do a lot of the things we do. And we're able to leverage that with other funding support through Project Green or the US Wheat and Barley Head Scab initiatives, some support through Michigan Craft Beverage Council and company trials that we run. But honestly, without all those parts, we really wouldn't be able to do what we do. All right. So wheat viruses in Michigan. So Dennis had pointed out some issues a couple of years ago. So we thought we should do a sort of more systematic survey to really try and get a handle on what was out there. So we brought a student in, Keely, and she's done a fantastic job at this wheat virus survey, getting out about the state and collecting samples and getting organized and doing all the testing work that's involved. Just to try and simplify things, we've color-coded these viruses by the type that they are, and I think more importantly, the vector host, or the vector that moves these things around. So at the top there, we've got barley yellow dwarf and cereal yellow dwarf virus, and they're vectored by aphids. There's some other viruses, soil-borne virus, wheat spindle streak, and that's vectored by a soil-borne fungus. Broom mosaic, and it's not really known exactly how that is moved around, so some mechanical transmission. Wheat streak mosaic virus, and another related one spread by wheat curl mite. So the reason that we picked these particular viruses is based on data from around the Midwest in terms of what people typically find, or some of the testing centers have been picking up. So we picked these particular viruses to test for. We ordered essentially like antibody kits to collect the wheat samples, grind them, and then test them for the antibody kit to see if it's infected with the virus. So it's really something of an involved process to go through correct identification. Here's just a few sort of classic examples of what things might look like. So barley yellow dwarf, typically that's looks like yellowing and purpling at the leaf tips is sort of a classic symptom for that. This is a mixed infection, so we've got a few different things in here: barley yellow dwarf, cereal yellow dwarf, and wheat streak mosaic virus. So that sort of complicates, diagnosis out in the field. And this is what we'd sort of consider somewhat classic for wheat streak mosaic. So streaking in that leaf tissue. So it can definitely be pretty hard to distinguish these apart based solely on foliar symptoms, and that's why we have to do that antibody testing back in the lab. And I should say with that, like if you have some sort of issue and you're not sure what it is, get it into the diagnostic lab where they can run these tests and confirm what the issue is for you. So the student did an excellent job collecting samples from across the state. And the way she did that was to base the amount of sampling intensity on the wheat acreage. So we had a lot more field sampled up in the thumb region just because there's more acreage up there. But she also tried to cover the state as much as possible wherever we had wheat. She collected or sampled 260 fields in 24 and 25 combined. So a lot of windscreen time. So this is just a couple of slides to summarize, really a couple of years worth of work. Across the top here is the 2024 data. So we've got wheat streak mosaic virus, barley yellow dwarf, and cereal yellow dwarf virus. And this bottom line is for 2025. I've just put the vector back up there to remind us. So this wheat streak mosaic virus is spread by that wheat curl mite. And then these other viruses here are spread by the aphids. Okay. One thing we did notice when looking at the data is that there appear to be some differences by year. So the color intensity is telling you how many fields or samples were positive in that particular county. So you can see for wheat streak mosaic virus, we certainly had a lot of it in 2024, but the amount was considerably higher in 2025. The opposite was true for barley yellow dwarf and cereal yellow dwarf, so they were certainly widely distributed and pretty highly prevalent both years, or both viruses rather. But in 2025, the amount decreased pretty significantly when we went and looked. So this suggests that maybe there's some differences in that vector activity, either that wheat curl mite having a little bit more activity in 2025, and inversely with the aphid, maybe more activity in 2024 versus 2025. There was some other viruses we looked for. They were pretty low abundance in the survey, so I won't go into them in a bunch of detail, but we looked for those, a pretty low abundance. Let's probably not spend too much time on those. So takeaways from the survey, it was pretty easy to find viruses out in the fields across Michigan. So in total, across those 2 years, she collected over 1,000 samples. So for every field, she would collect 4 samples, and they were sort of composite samples, so 20 leaves per sample collected 4 times across the field. So however we break it down, based on number of fields, about 90% of the fields were positive, or if we look at it at the county level, around about 91% of those counties were positive for at least one virus. So certainly a lot of it out there. Wheat streak mosaic virus was the most prevalent virus that we found out and about the state. And so that's led us to do some follow-up work with that particular virus. And on campus, we've also set up some field trials where we're screening a whole bunch of different varieties for their susceptibility to this virus. Keely did some greenhouse work, and what she used here is Ambassador as our susceptible check. This is a visual rating that she did, so the higher up here, the more virus symptoms that she was seeing. She used a variety out of Kansas with some resistance to this wheat streak mosaic virus, and just screened a whole bunch of different varieties. This is somewhat preliminary. We're doing field screening and some additional greenhouse screening to confirm these results. But it looks like we do have some differences in susceptibility across the varieties that we grow. And that might be very important as we move forward. So what are we doing now? So some of those varieties and a bunch of the yield, what would you call them, Dennis? The varieties that we got from you.
In the variety trial?
Yeah, just a variety trial, right? Anyway, we got from Dennis. Dennis helped us out and planted with his crew the variety trial set of lines, and we planted those out on campus. And what we used is a volunteer wheat field, and it had it had a lot of volunteer wheat, maybe not excellent, but a lot of it. And we basically went in and tilled a strip, and then Dennis and his crew came through and planted all these differential lines in like 3-foot long plots so that we can hopefully get really good virus transmission into those to assess this coming season. So I'm pretty excited about that. The other thing that's very challenging here is that I can take that wheat streak mosaic virus, and I could come to your farm if you wanted, and I could inoculate plants and demonstrate to you that, yeah, we can lose 50% of yield, right? So we know it can, and you guys know probably that this can be a problem. One of our big issues is though, like if there is some yellowing out there, what impact is that having? Is that impacting 1-2 bushels? We really don't know, and there's really nothing out there in the literature to look at these lower levels of infection and what does this really mean? We found it in a lot of places, but what is the impact? So we're hoping to do some microplot type studies to look at impact of these sort of minor infections. Are they significant? Should we be concerned? Should Eric and Dennis be focused on trying to breed that next variety that's got better resistance to these wheat viruses? So in terms of management, one of the most important things is trying to manage volunteer wheat. So we really don't want to have volunteer wheat across the laneway from our next planted crop. We got burnt on campus with some stuff that Kurt Steinke was doing next to us. He hadn't terminated his volunteer wheat, and sure enough, we had wheat streak mosaic virus come across the laneway and experience this similar type of phenomenon. So it's really important. The wheat curl mites may certainly be carried long distances on wind, but the majority of mites and virus transmission is going to have a very, very strong field edge effect, and that's typically what we see. So anyway, that's one of the main sort of takeaways here is don't have volunteer wheat next to your next season crop. The other important thing to emphasize, and Chris DeFonzo's here today too, so she can correct me, but we really don't have any good miticides or insecticides that we would recommend, at least, for controlling the aphid or the mites. So trying to manage the insect vectors is really not an option either. Again, it's really got to be about terminating that volunteer wheat and not having that in really close proximity to our next crop. I had some questions this morning. I was at another meeting about using wheat as a cover crop, and that's great, but I think, again, we need to be mindful of where you're putting that, and are you increasing the risk, because now you've got more wheat out there and maybe some volunteer wheat or cover crop wheat next to a next season's crop. So I think we really need to be mindful of that. Does anybody have any questions on the wheat virus work that we've been doing, or comments? Yeah. So as far as we know, rye doesn't seem to have the same effect. It doesn't seem to be as an effective vector for these viruses. I need to do some more digging in that space, but yeah. So we're certainly concerned about using wheat as a cover crop. Yeah. And no doubt, ditch banks and whatnot contribute some of the infections that we see out there, but I'm not telling you to burn all those out or anything either. But again, just being really aware of that volunteer wheat, because that's going to be your primary source. I know Chris and Anders are pretty keen to try and find these mites, because we haven't seen them yet. They're out there, supposedly. Oh, okay, so Chris is telling me they don't survive cold temperatures, so we might be, it'll be interesting to see what happens. Yeah, this season we might not have as much. So I think we've got a lot of questions about fall transmission versus spring transmission. The wheat curl mite can also survive in corn. We've got a whole bunch of questions about this system we're trying to figure out. And there's actually like a national movement here as well. Kansas called a meeting together to try and get a better handle on this virus issue.
Does the virus overwinter in Michigan on residue, or is there any management things that can be
Right,
done
thanks,
in the
Dennis.
off-season?
Right, so does the virus overwinter? So it won't overwinter on residue. It'll overwinter in that wheat crop, right? And this is where we're curious about the fall infection versus spring infections and when is movement really happening and transmission. But yeah, if the crop is dead, if you've sprayed it, that's not going to be a vector. It's going to be living. So it'll overwinter in that living plant. Has the potential to do that. Yeah.
How was the samples selected?
Yep.
Or was it like fields that had yellowing? Or?
Yeah, so how were the samples selected? So basically Keeley called a whole bunch of you guys and a whole bunch of co-ops and whatnot and was given permission to go to different fields. So yes, so Keeley is a good point. Keeley did go looking for symptomatic plants. So we could have done a sampling where we just walk around blindfolded and pick leaves, right? We probably wouldn't have found as much, but our primary goal here was just to figure out what was out there. And as I said, we know, like in that photo here, like that's a problem. You're going to be losing yield, right, when there's so much virus load there. But when there is some yellowing, what is that doing? We don't really know. And this is, despite these things being around for many years, there's not a good study to look at that at this point in time. But that's how we did it. We intentionally looked for yellowed plants and they were sampled. Yep. Thank you. All righty. Let's get on to this other thing I wanted to talk about. So I think this is kind of relevant too with commodity prices and maybe thinking, do I need that seed treatment? Should I bin run seed or not? So smuts and bunts have been a really big issue in the 1940s, 50s, before we had good seed treatments. Now with seed treatments, we're able to do a really good job at managing those. I just wanted to mention a couple in case you're out scouting and looking for things. So this is loose smut. So this doesn't seem to be one that we're particularly concerned with. We see this in our fields occasionally. This is pretty closely related. The fungus is pretty closely related to corn smut. It's a different thing, but it's closely related. Again, this is not really a big issue as far as I can tell. It's it produces sort of loose smutting on the outside of the head. So if you run your hand over this one's on there. I can do this, that's fine. Thank you. Right, so I just wanted to point out the two different types of smuts if you're out there sort of scouting and looking at your fields. So again, this is loose smut. I don't think it's as big a concern. If you were to run your hand over it, you'd pick up those black spores on your fingers, okay? So loose smut, different, caused by a different fungus. The one that we're more concerned with is common bunt or dwarf bunt. And in this case, the bunted grain is contained in that ovary tissue. So the fungus gets in there and replaces that ovary tissue with its own spores. And at harvest, this is what you'll see. So the seed is replaced with those spores that are inside of that seed coat, essentially. Okay. So under the microscope, this is what they look like. There's a bunch of work that's been reinvigorated around the system, I guess, just recently. For the longest time, we would use microscope images to look at these things and try to tell things apart. There's a couple of different species, and you guys might not care, and that's totally fine, but it does matter in terms of trade. So common bunt is one particular issue, and then we've got dwarf bunt, and China put some regulatory things in place back a few years ago now, 10, 20 years ago, with respect to dwarf bunt. They've relaxed things a little bit more now. So part of what we're trying to do at the moment is figure out which particular species are causing problems in Michigan so that we can then look at management practices. And we've seen statewide issues. We've seen issues up in the thumb, southeast Michigan, northwest Michigan. In northwest Michigan this last year, we had some considerable damage up in that part of the state. So these common bunt or stinking smuts, these sort of two very closely related diseases. Once we have infected heads, they go through the combine. Back in the days when this is a really big problem, you could even get explosions because of the amount of spores that get thrown into the air. So you could have fires because of that. But anyway, those spores can be thrown back out the back of the combine, fall on the ground, infect your soil so that you have that problem in the future. The other really big issue here is that we have then that seed becoming infected with these bunt spores. And the way that they infect is once those plants start germinating, they'll infect the plant and then grow systematically through the plant and up into the head and replace that ovary tissue with their own spores. And then, of course, the cycle continues. So again, more detail than you probably care about, but this is part of what we're trying to sort of figure out which of these species is our primary concern in Michigan. There looks to be some differences between this common bunt or stinking smut. And by the way, if you're not familiar, this produces that off fishy odor in grain. That's an issue, right, for processing. There's potentially a couple of species that are associated, and they have a slightly different temperature optima in terms of when they germinate versus this dwarf bunt. I mentioned, China had some trade embargoes placed previously. They've relaxed those a little bit, but they still test samples and make sure that there's not too many of these spores in there. So they'll look, and if it's above a threshold, then that might be a problem. Anyway, so there's some differences here, and we're trying to figure out what's really going on. Looks like MSU has sort of been involved with smuts for quite a while now. So this is a publication from 1918. Here's a couple of guys using some formaldehyde without any respiratory gear here, mixing it through the grain. There's kind of an interesting sort of note down here about, people thinking a little bit's good, so why not double it up, right? You guys might be familiar with that adage. Yeah? Yes? I'm seeing some smiles. So the point here is, don't do that with formaldehyde because you're gonna hurt your wheat. Anyway, pretty interesting. So there was quite a bit of work done some years ago, some in Canada as well. So we pulled some of the different publications that are out there. It's got a whole bunch of details here, really, but what we're looking at here is they used some bunted grain, they treated it and then planted it. And they had in their non-treated check about 9, was that bunted kernels, number per gram, so 9 bunted kernels per gram. But when they use some of these seed treatments, they are able to dramatically decrease that infection rates. So I guess this is a quick point to point out that seed treatments might not be silver bullets and completely eliminate a problem. If you start with dirty grain, you might reduce the issue with a seed treatment, but you might not eliminate it. And so that gets us back to thinking about, bin run seed. Should we do it or should we not? Well, it's just one of those decisions we have to sort of, think about. But that would be the advantage to using certified seed. At least it's been through the certification process. It should be free of this particular disease. So we're interested in re-examining these seed treatments. So a lot of the work was done, 20 years ago or so. There's definitely been a shift in some of the treatments that are available. So we're trying to do some work at the moment on campus to look at these different seed treatments. The short of it at the moment, for smut bunt control, use a seed treatment. We want to make sure there's a DMI fungicide in there. Difenoconazole seems to be a really good one, but there's probably others that are quite good. Have seed professionally treated. If you're doing a planter box treatment, you're probably not getting very good coverage of that seed. If you're not getting good coverage, you're not going to do a good job at really controlling any of that smut that might be on those grain. And of course, if it's known to be infected, we really shouldn't be using it as seed or cover crop, because that's just going to create issues in that field that you're planting with that infested seed. And this can survive in the soil for many years, and it's just something for us to be aware of as we think about this issue. All right. Are there questions on the smut or bunt? Yes, sir.
Do you have any comment on, what was it, last year, the Chinese nationals and U of M and the FBI?
Yeah, I did a bunch of interviews for that. So most of you probably know what that's about. So there was some University of Michigan, thank goodness, right? Not us. Chinese nationals that had brought in some fusarium strains to work on in the lab at University of Michigan. I don't think any of that was nefarious. I don't think there was any ill intent there. I think what happened is that they just skirted some of the rules and regulations that we're supposed to follow. So if I want to bring in an infested sample or fungus, even across state lines, I'm supposed to get a permit, which sort of doesn't make a lot of sense with fusarium, because how much grain are we carting across borders and whatnot, right? But I understand the reasoning, right, is to minimize any potential pathogen escape. So basically all they did is just skip that. They tried putting it in their backpack, and there's a few other little things that they tried bringing in, and they got caught and got in big trouble because of that. Yeah. So
Can it be a concern for us that?
Oh, right. Potentially. Bioterrorism is a thing. Yeah, for sure. In this case, though, I don't think that that's what they were trying to do. I think they were just interested in some of the molecular mechanisms of the way these organisms behave. I don't think they were trying to do anything. But yeah, that's why we have these regulatory processes and I guess border patrol checking bags. I know going to Tasmania, I sweated a lot. Going to Australia, I sweated a lot more, because we're even more particular about, because being an island, we're a lot more freaked out about bringing things in. But so you're far more likely if you go to Australia to get pulled over and just do some additional baggage screening and questioning than coming into the US. But it happens coming in as well. Yeah, I think one of the other big problems is all the trade of products. Right, there's all sorts of pathogens that get moved around. Right. So anyway, it's a good point though. Yeah. All right. So maybe let's move on, talk about some foliar disease management. Here's just a close-up of our research plot sprayer. This is really helpful because we can put 24 different products in there in a bunch of 3-gallon canisters. We've got 24 booms that are all GPS tripped, so as we're driving across, we can look at a whole bunch of different products in different experiments out in the field. So of course there's a lot of foliar diseases that can certainly impact yield. So it's good to stay on top of those and to minimize those where possible. Let's talk about stripe rust in 2024. And this is just a good one to keep on our radar in terms of thinking about fungicides and fungicide timing. What was really interesting in 2024 is that we had some wheat that was really late planted, and we didn't seem to get anywhere near or any detectable stripe rust, as opposed to Kurt Steinke's trial that was only a quarter of a mile away. He was planted on time and had a much better looking canopy, and he ended up with a lot of stripe rust throughout his plots. I asked him about fertility because we always get the questions, or maybe the plant just needs a little bit more of something. And he saw no effect from the fertility treatments he had in terms of managing this stripe rust. He had so much infection you could see the spores down there on the ground. Anyway, so I was really excited by that, being a plant pathologist and trying to test products and kill plants and things. So I went in and collected a whole bunch of leaves from his field trial. I basically put like a drop of soap in there, mixed it up, and that orange water is because of all the spores that were in there. So it was loaded, right? Just these few hundred leaves. I have a pivot on the farm on campus there, so I ran the pivot across our wheat just to get it wet, and then I backpacked this spore suspension on, and then we had some fungicide trials out there that we looked at in terms of how were these different fungicide products controlling stripe rust. So here's what happened. Here's a non-inoculated plot, so I did not put any of the spores here, and here's a plot that I had inoculated 18 days earlier. And you can probably tell that this is quite yellow compared to this plot that's much healthier. So we had some pretty incredible stripe rust take from that inoculation procedure. That was really helpful because it lets us look at different fungicide products. So we had a couple of trials out there. This is one where we were looking at foliar products. We sprayed them at that Feeks 8, that flag leaf just emerging growth stage for most of these products. And then one of them, this Prosaro, we put on at flowering. So we had sprayed with the fungicide at that Feeks 8 and then put the stripe rust out there about a week later for these particular set of treatments. And I've got our non-treated check at the very bottom there. Blue bar is yield, so I've stacked it by yield, and red bar is the amount of disease. So we had a lot of disease in that non-treated check that had no fungicide treatment. So it was really helpful because we were able to have a look at these different fungicide treatments, see how they were performing. There looks like there's some differences between the products. And what I would say is never use one trial's worth of data to make product selections. I like to look at multiple trials and look at what my colleagues are doing in other states too to see if things are trending out, because there's always some variability. And then that Prosaro, it looked the best, but this was also put on a week after the spores were put on. So I get the question, well, when should I put the fungicide on? Well, you need to put it on to control the disease, right? If you're going out preventatively, that's fine. But if the disease doesn't come in until later, maybe you needed a later fungicide application. We'll talk about timing in just a second, but fungicide timing is a really important part here. And here's a photo from even back in 2016. So we had another stripe rust epidemic back in 2016, if you can remember that. Here's our non-treated check. Here's our plot that we sprayed at that Feeks 9 growth stage, so that full flag leaf out growth stage. And what we observed is if we had gone ahead and made a prophylactic treatment ahead of time at that Feeks 5-6 growth stage, it was really too early to control that stripe rust epidemic because we put the product on. Products, they do a good job for a couple of weeks and then they start, waning in their performance. If we waited until flowering, it was too late in 2016, right? We had to be on there at that flag leaf growth stage. All right, so let's talk a little bit more about timing. We can come back with questions about that stripe rust. So let's break it up into T1, T2, and T3. So T1 being Figs 5, 6, T2 being that flag leaf, and T3 being that flowering timing. So partly I did this because I was really curious about this T1 timing, because I hear like, oh, we're going to go across the field anyway with a herbicide, so we're going to spike in some fungicide. Okay, that's fine, but why are we doing that, right? So we're curious about the yield impact from that T1 fungicide and these other timings too. We took data from like larger plot work that Martin Nagelkirk had done up in the thumb here, Kurt Steinke's fertility program at this farm and back at campus, and then a lot of our work as well from here and also back on campus. This was the spread of what we saw. I know you can't read all of these, but this is just the different timings. The positive response relative to no treatment, and the few where it appears to be negative, right? But the vast majority were positive. So let's look at it by the actual timing. What did we see? I was somewhat surprised by that T1 timing. On average, we were seeing around about a 4 bushel response compared to the non-treated check. I was surprised by the size of that. I know I've had a bit of argument with Wheat Pete about it. They don't tend to see as big a response as that, right? I think they claim about 1.5 bushels. I thought that was pretty interesting. That T7, or T2 rather, that flag leaf, about 7 bushels. That T3 at flowering, about 7.5 bushels. And combined, that T1 and T3 were at about 10.5 bushels, right? So anyway, I was quite surprised by that. It doesn't mean I'd necessarily recommend we always do that prophylactic treatment, because the more we put these treatments out there, the more likely we are to select for fungicide resistance. And once we get fungicide resistance, we're going to run out of tools to manage disease issues when they really pop up as a major concern. But, I mean, from a business standpoint, it's good to know the numbers. So I guess just another couple of comments about this. You're not guaranteed to see this response either, right? This is the average from a whole bunch of different trials. I think it's really interesting. Kurt Steinke and I got very excited about this and ran a bunch of trials here and then also on campus. And we did a 3-way combination where we had T1, T2, and T3. And we happened to hit low disease years and got nothing out of it, right? So anyway, it's just something to, food for thought, I guess. Any questions on that fungicide timing and yield and what we're seeing? Question? Oh. Yeah.
I noticed on the labels for fungicides, they give a heck of a range of application rate. Sometimes double,
Right.
Is there a way for us to quantify or qualify how much we really should be putting on?
Right. So the question here is on the label, the fungicides typically have a pretty wide range. Sometimes it can be like a 2x rate from your low rate to your highest rate. When we do our testing, we try and pick what is typically recommended by industry. And so we have fungicide efficacy charts, and when we put those together, that's based on testing, not at the highest rate, because that might not be economically viable, but at what is typically recommended for that particular product. We struggle with this too, like we can't test all the rates, but what should we be testing? So we typically test what is typically recommended. Industry. Yeah, what? Yeah. Yeah. Oh, companies, and then that would be pushed down to co-ops, manufacturers, and then pushed down through the manufacturing companies down through co-ops and whatnot. Yeah. But it is definitely a challenge because we have the same problem. How are we going to rate these different products next to each other when they got different label rate ranges? We struggle with that too. Mm-hmm. Any other questions on this fungicide work? I was going to just touch on head scab really quick. All right, let's chat about head scab really quickly then. So I guess it's really just a transition slide. So of course, head scab, that's the same fungus, that Fusarium species, can also cause that gibberella ear rot on corn. You guys are all familiar with that. We're doing quite a bit of work on trying to understand what species are out there on our wheat and on our corn for better management of both. We had this fusarium risk tool. Here's a snapshot from May 29th for 2025 showing some risk popping up just in that part of the thumb there. For the most part, we had pretty low risk for most of 2025 for head scab. I did want to put this up because we're developing tools on the crop protection network. And so one of those is bringing in this wheat head scab tool. So this same model, I really like this particular map and the colors and whatnot. I really don't like this pin system, but we had this, that's what we have at the moment, and I'm fighting with my colleagues to just turn this into a colored map. But what is up there is a whole bunch of other models as well. So if you're curious about soybean white mold, gibberella ear rot, we have a new model that's going to go up there. We have the wheat head scab model as well. And it shows it's the same model that's running on that other head scab site as to what we're putting up here in the crop protection network. And at this crop protection network too, we have all those fungicide efficacy guides. If you want to look at what products we have given different ratings for different diseases, and I think we have the rate that we tested at. So we have the label rate range, what we test at, and then how we rank these as excellent, good, poor or fair for disease management. Anyway, so this is a really helpful website. It's basically where I put a lot of my extension energy and outputs because it's a collaborative effort and it just makes it so much easier than trying to do everything alone. So in terms of head scab management, we've got a few different products now. So we've had Prosaro for quite a while, Tilt and Folicure, and there's generics of these too. There's your rate range rate for some of these. And for the most part, let's touch back on fungicide resistance. All of these one class, DMI type chemistries. We do have a couple of products, Miravis Ace and Prosaro Pro, that include a different mode of action in the products. And so partly that may help mitigate fungicide resistance development. But the more important thing is how often are we exposing these different pathogens and diseases to these chemistries. In short, this is what we've seen. So these newer products are performing at least equivalent, if not better, than the old products such as Carumba and Prosaro. And just my quick fungicide resistance slide. So we are certainly concerned about fungicide resistance when I go and talk to my colleagues in horticultural crops where they go out and make a lot of sprays, or in greenhouses. This is a massive issue because they are making multiple applications every year, so they're selecting for resistance. And so essentially what happens in the disease or pathogen populations that are out there, you may have an individual that's resistant. So when you make that fungicide application, you're selecting for that resistant individual, right? So the more we make those applications, the more likely we are to see a propagation of those resistant individuals. So in the lab, we are doing quite a bit of testing, and so this is typically how we do it. We culture the fusarium or whatever it is we're interested in. We do some measurements, and now we're using some AI techniques to sort of do some of these measurements, because we would, in the past, use calipers, and it's pretty time-consuming and not particularly easy, especially with these sort of funky-shaped growth that we often get from some of these species. So we're using some AI image recognition to look at how much things grow when we put different amounts of fungicide in the plate. And from that, we basically look at, we determine something called an EC50, so how much of the fungicide do we need to limit that growth by 50%? And by doing that work, we're able to look at, is there any fungicide resistance now, or is it indication that that's likely to be a problem in the future? So we've got a large collection of isolates, fusarium at least, from across Michigan, and we're doing some other work now with the Head Scab Initiative across the country to look at fungicide resistance. I just wanted to sort of show you that. I know that's, it's pretty basic stuff. You guys might not care too much about it, but it's important that we're out there checking for any resistance issues that might be developing. These graphs are just showing us for the how many of the different fusarium isolates we have and how sensitive they are. So the further to the right of here they are, the greater the concentration that they're able to grow at. So these are the isolates that we're concerned about in terms of overcoming the fungicide tools that we have. And I guess part of the issue there is we really rely pretty heavily on 3 modes of action for a lot of the field crop diseases that we work with. So there's certainly concern there. But anyway, I should have ended on a more happy note. Does anybody have any questions about the foliar fungicide work or any of the fungicide sensitivity work we're doing? Or anything else? Nope. Yeah.
You had a chart 5 or 6 back. You went through it really quickly.
Yeah.
It showed Spirax on the right in green and everything else in red.
Yeah.
Explain the ABCs.
Yeah, sure. So I pulled this graph. This is a multi-state effort. So this is data that my colleague in Ohio pulled together. So we do these trials here. They're done in Ohio, Indiana, other places, and we pull all that data together and then look at what the trends are, right? And this is just the statistical differences. So this is the amount of head scab. So in a non-treated check, how much head scab on average across all those different trials we're seeing. And you can clearly see that most of these products are doing a good job. So what we look for is statistical differences, right? If they all share the same letter, then we can't technically say that they're different. So I can't technically say that Miravis ACE is different from Spherex because they share a letter code, that's C. But that Miravis ACE is a little bit better than Carumba and Prosaro, right? And same, I mean, you guys probably care more about the actual DON. So here's the DON data, that vomitoxin data. So non-treated and our different treatments, right? Yeah, no worries. I thought about putting up some of our own Michigan data from last year, but it gets, I don't know, it'd take a little while to really go through it and make sure you guys understood what was going on. But yeah. Yes, sir. I mean, I think in general, one is as good as the other, and some of our data seems to show that Miravis ACE is perhaps a little bit better than the others, than Prosaro Pro and Spherex. And I think I have hidden slides there. I was toying with playing, but making sure we all understood, we're all on the same page when we walk through them. But yeah, a little bit better. We've also been looking at like double applications, coming through with the Miravis ACE and then coming back through 4 to 6 days later with another product to see if we can drop that head scab and vomitoxin even further. And it shows some promise doing that. Obviously, it increases cost. You guys don't want that, but we're just looking at other ways to help try and mitigate these issues. The other thing I was playing with too. So we pretty often select a susceptible variety and a resistant variety. And that's where there's a very big difference in the amount of vomitoxin that accumulates, right? So we'll use Ambassador as our susceptible check, and that tends to get lit up. This last year we used Wharf as our like resistant variety or partially resistant variety, and that had probably 5 times less vomitoxin than that susceptible variety. So I think this is a really important point. Variety selection is absolutely key. And we've come a long way over the last 20 or so years, in part through the head scab initiative and the breeding efforts that have been going on. There's still weaknesses out there, of course, but things have definitely moved forward. But in terms of just practicalities, think about variety selection and know what your perhaps weaknesses of the varieties are. So if you're growing a variety that you really like, great, but is it susceptible to stripe rust? Just know that ahead of the season so that if we have stripe rust starting to show, you might have to get out there and make that timely fungicide application to manage that. Yeah.
Marty, I have a question.
Yep.
With our below normal temperatures we've had the last month, does that have anything to do with disease pressure
Right.
this year?
Yeah, so, I mean, Chris and I were sort of chatting about that, right? So insect-wise, I would say yes. Maybe... From the pathogen standpoint, though, like we store a lot of our isolates at minus 80 Celsius for long-term storage. If I want to like keep something alive for a long time, we'll put it in the deep freeze. So, right, yeah.
Okay, any more questions?
Thank you.
Grab something out of the basket there if you
Thanks,
want
man.
to.